How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me are all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hello, you're listening to DNA Today, a genetics podcast and radio show. I'm your host, Jenna Guiltinan. DNA Today informs you on what's happening in the genetic world. During my broadcast, I educate you, the public, on genetic and health topics through event coverage, news stories, book movie reviews, and interviews. Guests include genetic counselors, like today, researchers, patient advocates, and professors in the field of genetics. Joining me today is Jenna Guiltinan. She is a laboratory genetic counselor who received her master's in genetic counseling from California State University Stanislaus in 2013. She worked at Ambry Genetics as a reporting genetic counselor for over 4 years. However, she recently left her position to pursue a new laboratory position. And so as a laboratory genetic counselor, she helps interpret and report the genetic test results, and we'll kind of get into what that means and how you go about that. So thanks so much for coming on the program.
Yeah, thank you so much for having me.
Let's start out with what genetic counseling is for anyone listening that hasn't heard of it or they're not really sure exactly what it means.
Genetic counseling is the process of meeting with patients and sometimes their families to discuss the risk of either having a genetic condition or passing on a genetic condition, and talking about the options available for patients in regards to testing for the condition and family planning and management for that condition.
So genetic counselors are kind of aiding in teaching all of this really complex information down to the education level of the patient.
Yes, it definitely involves a lot of education and describing complex medical terminology and ideas to patients in a way that they can understand it, because the only way to make informed decisions about these genetic conditions is really to understand the options and the condition itself. So being able to explain things in a way a patient can understand is very important to genetic counseling.
And so kind of what you were talking about is a traditional role that we say, quote unquote, which is becoming more kind of untraditional, I guess. Other roles and subfields in genetic counseling are popping up, but for now, I guess it's still called traditional genetic counseling of actually meeting face-to-face with patients or sometimes through the phone. But there are different subfields in genetic counseling, and one of which is laboratory setting, where you actually work.
Mm-hmm. Yeah, exactly. So the traditional genetic counseling, which is generally face-to-face contact with a patient, is, like you said, still kind of considered the traditional role. But as technology is increasing and genetic testing options are increasing, there are a lot of different roles for genetic counselors like myself. So when I graduated in 2013, I actually took my first job was with Ambry Genetics as a reporting genetic counselor in a lab. So I personally, other than the time I spent in school in my graduate program doing clinical rotations, I've never actually met with patients as part of my job. So I've always been on the other side, on the lab side of the process.
So as a lab genetic counselor, what do you do? What are your roles if you're not meeting with patients?
So there's a lot of different things people trained with a genetic counseling degree can do at the lab. I personally, as a reporting genetic counselor, my role involved basically interpreting the genetic test results. So the lab that I work for offers many, many different types of genetic testing, anything from multi-gene cancer panels, so up to 50 different genes that are all associated with an increased risk for cancer, to even whole exome sequencing, which is where we essentially sequenced all of the genes in the human genome. My role would be to interpret the results of those tests. And in order to do that, you look at the information that was sent in from the ordering physician about the patient. So you look at their clinical history, you look at what we found, and you essentially try to make a call, or a classification. So we wanted to be able to give a result that would be helpful to the provider who ordered the test.
And in this role, you're kind of taking all of the work of the lab technologists and different people that have worked with the samples before you, and you're taking all that data and kind of making it a little bit more English and less just data?
Yeah, exactly. That's actually a really good way to say that. So after the test was run through the lab, we have the actual genetic test result, which is something that is written in specific terminology, and it can look like gibberish to a provider. So what we do is we would write a paragraph explaining what that actually means and what it means for the patient and how you can use this information for management.
And a lot of providers, you'd think, oh, everybody, if they're in healthcare, they understand genetics, but that's not really the case. You have to have, genetic counselors have 2 years of master's level training. So it takes a lot to be able to break down the information to the point where you can do that.
Yeah, that's right. And because genetic counselors these days are actually in really high demand, not all patients get to see a genetic counselor before they have a genetic test ordered. So sometimes we would see genetic tests being ordered by all different types of medical providers, not just geneticists or genetic counselors. So that made it even more important that the results were explained in a way that whoever ordered the test could understand it and then share that with the patient.
And as you said, genetic counseling is just rapidly expanding. In Connecticut specifically, it's the third fastest growing job. But that really expands to a lot of different states in the United States, but also in the world, that genetic counseling is becoming much more, as you said, in demand, that there's a lot of job openings and not enough genetic counselors for them. So in terms of laboratory genetic counseling, why do you think a lot of genetic counselors are wanting to, join in this laboratory setting as opposed to starting out counseling patients or just hoping to continue counseling patients directly? What is the draw to joining into the laboratory setting?
I mean, that's a really good question. I think there are several reasons. One of them might be the freedom of time that you get. So I didn't really word that right, but I guess what I'm trying to say is there's a lot of flexibility in with labs. So I know that specifically for the lab I worked at, there was the option to work remotely. So that was really important for a lot of people who had a family and maybe they had young kids and they wanted to be home and be able to kind of log in and work when they could, and then be with their kids or take their kids to doctor's appointments or parent-teacher conferences whenever they were needed. So the remote positions, you often have a lot of flexibility in the hours you work, when you work. Obviously, you don't even have to leave the house to work. So I think that's a big draw.
Yeah, I work from home, and I gotta say, I love it. I'm going to be a little sad when I don't get to work from home anymore. So that's definitely a big perk.
It's definitely a perk. It's really, really nice. And for some people who live maybe in more rural areas, that's really the only option for them because, if clinical genetic counselors typically work in more metropolitan areas where there's more patients or there's a geneticist to work under, and that's not always the case. But I think there are parts of the country that don't even have clinical positions right now.
Yeah, that seems to be the case, which is, why it's so great that with, Skype and different services like that, you can even video chat with a genetic counselor. And so you're, getting even more than what a phone experience would be of kind of having FaceTime, but, not a full kind of in-person experience.
Yeah, and there are laboratories that offer video counseling. So there are definitely laboratory counselors that do actual patient counseling through video. I just wasn't one of those genetic counselors.
Haven't hit that yet, but probably in the near future you'll end up doing something like that.
I thought about it. It was an option actually at the lab I was working at. It just wasn't something I was had the time to do at the time, but it did seem like kind of an interesting idea and maybe something I would like to pursue.
Yeah, we've had a couple genetic counselors on the show that have done that, or that's part of their private practice or something, to be able to have a wider reach with people, not just in their physical location. So it's definitely something that's helping the field advance. Technology is always kind of doing that.
Yeah, I mean, whatever we can do to get genetic counseling to more people, I think is great. Even though face-to-face is probably, preferred for everybody involved, it's just not feasible right now for so many people out there just because there's not enough genetic counselors. So whatever we can do to increase genetic counseling to the patient population, I think is an improvement.
Definitely. And increasing awareness like we're doing right now.
Yeah, yeah.
So another thing I kind of wanted to come back to is genetic variants. You kind of mentioned them beforehand, and having that be part of your report sometimes where you find a genetic variant. But what is a genetic variant?
So a genetic variant is basically anything in the genome of somebody that differs from the reference sequence. So it can be as small as a single nucleotide change, one of those ATGTs, maybe just one of them is changed, to whole regions of a gene or even larger than a gene that have been deleted or duplicated, or basically any change in the genomic sequence that differs from the reference sequence. Not all of these changes are bad at all. I mean, we all have hundreds and thousands of them. So I think that the challenge is when you find one in a genetic test, then what does that actually mean for the patient? Is it causing the patient's disease, or is it just totally a change that doesn't have any impact on the patient's clinical presentation?
And when you're looking at that and trying to decipher which it is, that if it kind of is neutral or, it has negative consequences or even positive, I suppose there's some of those, how do you actually start that process? What do you use to actually classify these variants?
So it's kind of like being a detective in a way, and that's actually why I love my job so much. It really involves gathering different types of evidence from many different types of sources. So we look at things such as the literature. So we look at published papers to see if there's anything out there in any of the hundreds of scientific journals where somebody has found the same genetic alteration in another patient that had a disease. We look for functional studies. So sometimes people can perform research studies in a lab, like looking at cells that have those changes to see if the cell is behaving differently because of that genetic change. We have tools called in silico models, which are like computer modeling programs that can try to predict whether that change is actually going to alter the protein. So there's a bunch of different sources and types of evidence. Another really important one is population frequency. So we look at all these open source population databases to see how common that change is, if it's seen in healthy populations, if it's been seen in sick populations. So there's a bunch of different types of evidence. We try to gather as much as possible, pull it all together, and then using that evidence, we try to make a classification. And I can get into the types of classifications.
So take me through, I guess, how many different classifications there are. And I don't know if there's like what kind of the differences are between them.
Yeah. So most labs these days use a similar classification system, and this is all based off of the ACMG guidelines, which is the American College of Medical Genetics, which is a group that's very influential in the field of genetics, and they have published guidelines. So most labs try to follow their recommendations, and they recommend a 5-tiered classification system. So a variant that is not expected to cause disease is benign, and then that would be class 1, and then it goes all the way to pathogenic, which is an alteration that is expected to cause disease. So those are the two...
the extremes.
The extremes, yeah, the ones with the most evidence. So if we are almost, 100% sure that this alteration is not going to cause disease, we would call it benign. And if we are almost 100% sure, because we have enough evidence to back it up, that this alteration does cause disease, we would call it pathogenic. And then in between, we have variant likely benign, what we call a variant of unknown significance, which is when we really don't have enough evidence to make a call either way for benign or pathogenic. And then we have variant likely pathogenic.
And for those variants of unknown significance, I've talked about this on the show a couple times, and just kind of they wreak havoc for genetic counselors and the people receiving results and patients. It's really hard to get something that you're like, we don't know after you go through all the genetic testing process. So what options are available for patients and clinicians when they get this variant of unknown significant result?
Yeah, so I agree. These variants of unknown significance are very frustrating for everybody involved. They're frustrating for us at the lab because we feel like we're not giving an answer. And like you said, they're frustrating for the clinicians and for the patients because you go through all this testing and then you get a result with basically just saying, we just don't know. We don't have enough evidence to say either way. So there are a couple options available. Most labs will offer some kind of family studies, often for free, where you can test other family members for that variant of unknown significance to see if they have it or not. And the idea is that you're trying to see if that variant tracks with disease. So if there are several family members that are affected and you test them all and none of them have that variant, then that means you have just gathered some evidence to show that that variant is not causing the disease in the family. On the flip side, if you have several affected family members and you test them all for that variant and they do have it, then you've collected a little bit of evidence to show that it might actually be pathogenic. So that's one of the options you have for families that's available at most labs these days, usually for free, but oftentimes you have to go through a process of applying.
A little bit more involved to get to that level.
Yeah.
You're testing multiple family members, so you got many samples happening.
It's definitely not easy.
Is that usually covered by insurance, or is that kind of a case-by-case basis?
I don't think that would normally be covered by insurance. It's oftentimes offered for free by the lab as part of variant analysis, but it's not usually, insurance wouldn't typically pay for testing for a variant of unknown significance.
And for patients that receive results that are a little bit more clear, that it's not, unknown significance, but maybe it's benign or pathogenic, how can they use this information to then make healthcare decisions and kind of be armed with this knowledge?
So let's use the example of BRCA gene, because I think that's one that's a lot of people are familiar with.
Especially with Angelina Jolie in the public eye.
Yeah, exactly. So Angelina Jolie had a BRCA mutation. I'm not sure if it's BRCA1 or 2. There's two different genes. I think it's
1. I'm not 100% sure, but I'll put that out there.
I think you're right, actually. That sounds right. So BRCA1, if you have a pathogenic or likely pathogenic mutation in the BRCA1 gene, then we know that you're at an increased risk for breast and ovarian cancer. So if you had a genetic test and you got a result like that, you have some management options available to you. One would be you have the option of having more mammograms more often and starting at a younger age, or even having a mastectomy before cancer to try to prevent you from having cancer, breast cancer.
Yeah, that would reduce your risk to almost zero. We don't really say hundreds and zeros in science, but pretty close to it, right?
Mm-hmm. Yeah, close to it. Close to zero, but not zero. So that's just for cancer genes, but there are different genes that cause all different types of conditions. So for genes that cause like hereditary heart conditions, sometimes there's the option of getting like a pacemaker put in even before you've ever even had an issue, just because you now know that you're at risk for some sort of heart condition.
And so with a lot of this, it's a lot of prevention that you're able to do because you know about something before it could happen.
Yeah, so depending on the condition, there are a lot of options available to try to prevent disease. So testing, if one family member gets a positive result, like a pathogenic result, then you can test any at-risk family member who's never even been sick before and try to prevent them from getting sick. So that's really the power of genetic testing. And the problem is if you have an alteration and we weren't able to call it pathogenic, we called it a VUS, then those options just aren't really available because we don't know if that genetic that change is what's causing the disease in the family. So testing the family members isn't really going to help you decide who's at risk. So in that case, you kind of still have to treat everybody in the family as at risk.
With more information, we'll kind of be able to identify a lot of variants of unknown significance as having significance and classifying them with that additional kind of data and being able to make those calls.
Yeah, that's the idea. I mean, we all want to be able to classify the variant. Leaving something as a variant of unknown significance isn't very helpful. So, yeah, with more data out there, with more people publishing studies, a lot of there's a big push in the field to share data, for labs to share their data online. And
hashtag free the data.
Yes, exactly. Because that actually really helps. Because if you say you see a variant in, you've seen it for the first time at your lab and you just don't have enough evidence to call it anything. So you leave it as a variant of unknown significance. But you look online and another lab has seen it, 10 different times. Maybe they've just gotten lucky or they've tested more patients. And if you're able to share your data with them, having that many more people who have had the alteration, it's just more evidence to be able to make a decision. So it is really important to share data. I will push the hashtag.
Definitely,
data.
Yes. We will use that promoting this episode. Now, a lot of this is obviously very kind of confusing, of going through and sifting through this data. How did you learn this? Did you learn this while you were a student in graduate school for genetic counseling, or was this kind of more on the job?
Yeah, so I didn't actually learn most of this in genetic counseling school. I learned it pretty much all on the job. So my first job at Ambry, I was trained in variant classification and techniques and the types of evidence we use. And it took a long time. I mean, they really invested a lot of time and energy into training me. And I really am appreciative of that, because I've just really loved my job and my career path since then. But I think now that more and more genetic counselors are going into positions like this, most schools that I'm aware of are starting to add classes or at least workshops in variant classification, so that graduating students are ready and prepared to start a career in a job that involves, like, a laboratory position that involves variant classification. And even if they don't go that way and they still want to be a clinical genetic counselor, I think having the background and the knowledge of what goes on at the lab can actually be really useful when it comes to counseling patients who have variants of unknown significance.
Because you know all the background of kind of how it works and what goes into it, and being able to actually look at some of the data yourself, right?
Yeah. Mm-hmm. Being able to check the databases yourself, so that you can, stay up to date and tracking that variant over time and seeing if there are any changes. Yeah, I think that's really, really useful. Yeah,
an important point you just said there is that variants can change. So it's good for patients to check back with their results and make sure that kind of the interpretation of it is still relevant today, because maybe 5-10 years ago it was a little bit different.
Mm-hmm. Yes, definitely. We're always constantly trying to either upgrade a variant of unknown significance or downgrade it so that it's more useful. And yes, so the classifications do change over time, and it is important for patients to stay on top of that. And we usually will recommend that a patient checks back in, maybe once a year with either their genetic counselor or the lab to see if there's been any changes.
Be an advocate for yourself and really be looking for those changes and, new things happening.
Yeah,
now,
definitely.
As we wrap up the episode here, I did want to ask you what changes in progress you see in laboratory genetic counseling in the next 5 to 10 years.
I think that we are getting better software tools to help us. I think that labs are hiring more PhD structural biologists that can help with variant classification. I guess I'm just seeing variant classification in general getting more complex, and we're getting better tools to help us do our job. I know that's kind of a vague answer, but...
no, it makes sense. Yeah, with more technology and more data, it can become more confusing and more organized kind of at the same time almost.
Yeah. And, I do talk about this. I was just talking about this with my my boyfriend earlier today. He asked if he thinks robots someday will replace our jobs. And I don't think they will, but I do think robots are going to, not robots, but artificial intelligence is going to make our job more efficient and easier and more accurate. But there's always going to be a place for actual genetic counselors in the lab.
Yeah, we can't really just have
clinic.
Everybody read straight up results, because that's going to be really confusing for a lot of patients and even doctors and clinicians.
Yeah.
Well, that about wraps up today's episode of DNA Today. It was a packed episode talking about a lot. Thank you so much, Jenna, for sharing your insight being a laboratory genetic counselor and all about these different variants and how we sort and go through them. Thanks so much again for coming on. It's really been awesome.
Yeah, it was definitely my pleasure. I know that this is really complicated, like dense field, and if anybody has questions, you're definitely welcome to reach out to me, and I'm happy to talk to any genetic counselors or students out there who want to become a genetic counselor and have questions about this.
Yeah, if you are on Twitter, you can connect with her at Jenna Guiltgc, and so she can answer you on there. And you can also find genetic counselors like her and others at findageneticcounselor.com. This is an awesome resource. This is what I started using when I was looking to get more experience in genetic counseling and have a few internships from it. So you can go on there, and there's even a there's a link for students, healthcare providers, or other individuals interested in speaking with a genetic counselor. So kind of look for that. That will be in the show notes as well. At DNApodcast.com. And I'm also on Twitter at DNA Podcast. And recently, new to Instagram for this show at DNA Radio. So that one's a little different, DNA Radio. But any other questions that you want to send in for me or Jenna, I can pass those along. info@DNApodcast.com. You can send them in. So thanks for listening, and join me next week to learn and discover new advances in the world of genetics.
You and me, genes of you, and they're all made of DNA. We're all made of the same chemical DNA.